Hello everyone, welcome to my Bio10 Ambassador Program. In this video, I'll introduce why some people can't drink milk with measuring lactase enzyme activity. Have you ever witnessed such a situation? Some friends can drink milk without any problems, but others get stomach pain immediately after drinking it. The problem here is not with the milk itself, but with whether our bodies can properly process the lactase in the food, from life to class. Let's introduce some basic knowledge for this phenomenon. In Bio10 course, we learned that all activities of life are based on metabolism. Metabolism is a series of interconnected chemical reaction networks that can convert the substances we consume into forms that the body can utilize. For example, catabolic pathways break down large molecules and release energy. Anabolic pathways use energy to construct new substances. It is precisely the catabolic process that enables us to obtain energy from food. All metabolic reactions follow the laws of thermodynamics. The course tells us the first law: energy cannot be created out of nothing. It can only be transferred or transformed. The second law: energy transformation often increases the disorder of the system, which is entropy. This means that any chemical reaction carried out by the body cannot be useless. It must be accompanied by energy change. When it comes to energy, we also learned a very important concept: free energy, which is G. So, if the delta G of a reaction is negative, it is a spontaneous reaction. If delta G is positive, additional energy is required to drive the reaction. But the key point is, even if delta G is negative, the spontaneous reaction does not necessarily occur quickly. Many reactions, also designed to occur, are hindered by an energy barrier, the activation energy. At this point, enzymes come to play. Enzymes are catalysts within living organisms that can significantly reduce the activation energy required for reactions, enabling them to occur rapidly while the enzymes themselves are not consumed. The characteristics of enzymes discussed in the course include: first, high specificity; second, combine with substrates through active sites; third, reusability and high efficiency. The presence of enzymes is a key factor enabling life to carry out various reactions at normal temperature.
Repeating the connection with real-life phenomena, among all enzymes, the one directly related to our consumption of milk is lactase. It is responsible for breaking down lactose into glucose and galactose, allowing the body to absorb and utilize its sugars as energy. If a person has insufficient lactase, lactose cannot be broken down. It then enters the large intestine, where it is fermented by bacteria, resulting in symptoms such as bloating and abdominal pain. These are the lactose intolerance symptoms.
Therefore, from the common phenomena of drinking milk to energy, metabolism, thermodynamics, and then to the activation energy and specificity of enzymes, and finally back to the activity detection of lactase.
All of these form a closely connected biological chain. Next, I will explain how we measure the enzyme activity of lactase in the experiment using the ONPG assay. Step 1. Extract the enzyme from commercially available lactase supplements. This is very interesting. Instead of using laboratory-grade pure enzymes, we simply use lactase tablets that are available on the market. In the experiment, we do the following steps. And the purpose of this is to simulate the actual state of enzymes in complex environment in the real world, and also to help us better understand how enzymes as drugs or supplements function in the body. For the step 2, why not use lactose but ONPG instead? The extra lactose and its products are all colorless, and you cannot observe their changes with the naked eye or a spectrophotometer. However, experiments require the ability to see the enzyme's reaction taking place, so we use a special substrate, ONPG. When lactase cleaves ONPG, a yellow product called orthonitrophenol is produced. The intensity of the color directly indicates the activity of the enzymes. The darker color means the faster the enzyme cleaves. For the step 3, we set different reaction times to measure the speed. The activity of an enzyme is initially how much product can be produced in a unit of time. Therefore, we prepared 4 identical reaction systems and terminated the reaction at different times, like this 4. At each time point, we add sodium carbonate to immediately determine the reaction, and this makes the color more pronounced, facilitating the detection. Step 4. Measure the absorbance using a spectrophotometer. We transfer the reaction solution after termination to a 96-well plate and measure the absorbance using a spectrophotometer at 420 nm. The higher the absorbance means the more yellow products, suggesting the faster the lactase reaction.
Returning from the experiment to life, just now in the experiment, we saw that ONPG turned yellow under the action of lactase, which is actually a very intuitive visualization. But the more important significance of the experiment lies in it helps us understand what various phenomena related to lactose occur in real life. The difference in color intensity that we observed in the test tube represents the molecular level manifestation of whether lactose can be digested in the body.
Lactase activity is not only related to whether the stomach acts or not, but also holds significant importance in medicine.
For example, newborn babies have a high level of lactase enzyme naturally, because breast milk is rich in lactose, which is an important source of energy. Some rare genetic mutations can lead to congenital lactase deficiency, requiring special feeding. Certain intestinal diseases, such as enteritis infections, can temporarily damage the small intestinal mucosa, reducing the amount of lactase. The expression of lactase in the elder population also decreases year by year. All of these reflect a fact: the quantity of lactase is not constant. It varies with age, health condition, and genetics.
This applies not only to lactose, but also to alcohol metabolism, drug metabolism, starch digestion, protein tolerance. Lactase is just a very typical example that, in the simplest way, tells us the body's capabilities are determined by the enzymes.